Thank you for coming to the talk. My name is Yafang. I work with Professor Cunningham.
Talk into the microphone.
Oh, sorry. Can you hear me now? Okay, my name is Yafang. I work with Professor Cunningham. Today, I'm going to talk about detecting protein and microcancer biomarkers using the silicon-based photonic crystals and the resonance-coupled laser scanning instrument. So this is the outline for today's talk. First, I will provide you with the motivation, and then I will show you how our photonic crystal enhanced fluorescence technology works. And finally, I would like to summarize my talk. So this work is motivated by the strong needs for personalized medicine. So personalized medicine is a highly customized treatment for each individual. So it is possible because our increasing knowledge about gene expression and protein biomarkers. So what can personalized medicine bring us? It can help us early identify the medical conditions before any acute symptom occur. It can help us select the most effective therapy without wasting any precious time and resources. And also, it can use the diagnostics to monitor the whole course of the treatment. However, personalized medicine could raise questions such as its cost effectiveness. So to reduce the cost and bring the convenience to the patient, we need the point-of-care diagnostics. So in point-of-care diagnostics, the non-professionals, such as general populations, will perform the test at some convenient places, such as the doctor's office, remote clinics, or even home. So that's why the point-of-care diagnostics could reduce the cost and bring convenience to the patient. To be qualified as a point-of-care system, you need the assay automation and the small instrument and low cost, and the data should be easily accessed. For cancer biomarker is important for the early diagnostics and personalized medicine for cancer. So cancer biomarker is a protein that is produced by the tumor or the body in response to a tumor. So by monitoring the amount of the biomarkers, we can detect the cancer. So the good news is that the biomarker can be found in circulating blood. So that means we can perform the blood test to detect the cancer. However, the bad news is that the concentration of the biomarker is extremely low. So the current technology is either too expensive or they are not sensitive enough to detect such low concentration biomarkers. So here we would like to solve the problem. So we would like to build a point-of-care system that can be used for the cancer biomarker detection. So it must have the capability of multiplex assay and have high sensitivity to picogram per mil detection limits. And also, it just needs small volume of the sample. And to be qualified as a point-of-care system, it must have low cost and has short assay time and small and inexpensive instrumentation. So how can we develop such inexpensive and small system? Here, we use our photonic crystal enhanced fluorescence technology. In this technology, we have the photonic crystal surface, which can efficiently couple the laser light into the fluorescence molecule, and meanwhile efficiently extract the fluorescence light from the fluorescent molecules. By this combined effect of enhanced excitation and enhanced extraction, we could obtain large fluorescent signal. Therefore, we could detect the biomarker with lower concentration. So how this enhancement works? So this is a 1D photonic crystal structure. It's comprised of the periodically changed low refractive index surface coated with the high refractive index material. When the light incident onto the surface, at the resonant angle, the transmitted light and the reflected light will constructively interfere with each other. Therefore, we can see the resonance. And at optical resonance, the local field would be enhanced. To verify that, we did simulation. So through simulation, we can see that the strongest local field can be enhanced by a factor of 100 times compared to the incident light. The only requirement is that the incident light must be collimated at a resonant angle. So we can see the enhancement factor, the enhancement effect through simulation. But can we see it also in experiment? To verify that, we first fabricate our device. So our device is on a silicon. We use the silicon as our substrate because the silicon fabrication is very easy and mature. So we can send our wafer to the outside foundry to etch the grating structures. And we cut it into even smaller pieces. So each piece just costs $1, which is totally affordable for the patient. For the low refractive index surface, we use the silicon dioxide. We use this material because it provides almost no fluorescence. Therefore, the background could be extremely low. So we would like the next step is that we want to see if we can see optical resonance on these fabricated devices. So to verify that, we measured the reflection spectrum of the device. So in the reflection spectrum, the peak means the optical resonance. So here, we measure both TM mode and TE mode, and we can see one peak for TM mode and one peak for TE mode. So by properly designing our device, we can utilize these two optical resonance. One optical resonance, which is the TE resonance, is aligned with the excitation laser light, and the TE resonance is aligned with the emission light. By properly aligning these two optical resonance, we can have the combined effect of enhanced excitation and enhanced extraction. To detect the fluorescence signal, we built our customized line scanner. So the light passes through the cylindrical lens and the objective and project to the photonic crystal surface to excite the fluorescent molecules. And the excited fluorescent signal would be detected by the CCD camera. By moving the translation stage of the laser, we could tune the incident angle of the laser. This is very important, since to achieve the maximum enhancement, we need to have the incident angle match with the resonant angle. Because we have the enhancement, the fluorescence enhancement, we don't need this powerful laser to excite the molecule or the very expensive CCD camera to detect the signal. Here, we just need the inexpensive laser and also the inexpensive CCD camera for the whole detection system. So the total cost of all of the components is less than 10K. So this price is totally affordable to a patient. And also the scanning speed is pretty fast. We can scan our device in 60 seconds. So the next questions are, can we use this? Can we see fluorescence enhancement when we detect biomarkers? And would the fluorescence enhancement be useful for the biomarker detection? So to answer this question, we did two experiments. One is that we detect the protein biomarker. The other is that we detect the microRNA biomarker. First, I'm going to show you the results from the protein biomarker detection. So to detect the protein biomarker, we performed the sandwich immunoassay. We first coated the substrate with a capture antibody. And then we add a blocker, and then we add the our sample, which contains the antigen. Here, this antigen is a biomarker. So this antigen can be captured by the capture antibody. And then we incubate and wash. And then we add the detection antibody, which can attach to the antigen. And then we wash, incubate and wash again. And finally, we add our fluorescence tag. So this fluorescence tag will also be attached to the antibody. So if there is antigen or the biomarker exists in a sample, then there would be fluorescence tag. Therefore, we could see fluorescence signal, and more antigen will result in stronger fluorescence signal. So that's how this assay works. In our experiment, we're using two biomarkers. One is IL-3, and the other is TNF-alpha. They are all breast cancer biomarkers, which can be used to detect the breast cancer. And we dilute our sample into 7 concentrations so that we can see what's the lowest concentration we can detect for both of them. And the antigen or the biomarker can be captured by the capture antibody, which I showed previously. And the capture antibody is contained in a tiny spots, which is printed by the deep pen nanolithography. So we have 4 kinds of spots. 2 spots one of them are the anti-TNF-alpha spots, which can be used to capture the TNF-alpha in a solution. And another is the anti-IL-3 spots, which can be used to capture the IL-3 in a solution. The other 2, one is a positive control, which always emits the fluorescent signal, and the other is a negative control, which always be dim. So here, this is a result. So to see if we do have the enhanced fluorescence effect, we did the experiment both on the glass slides and on our photonic crystal surface. So for the glass, we use the very expensive commercial scanner, which costs $200,000. So you can see this price is not affordable to each individual. And for our photonic crystal, we scan it with our customized line scanner, which costs only $10,000. So this is totally affordable. And here, this is the results. We can see that both of them have the same concentration of biomarkers. However, the fluorescence signal from our device has 20 times more fluorescence signal than that on the glass slides. So this verifies that we do have the enhanced fluorescence effect on our photonic crystal device. So what does this mean? How can this enhanced fluorescence effect benefit the biomarker detection? So here we plot the dose response curve, which shows the fluorescence intensity change as a function of the concentration. So from this figure, we can see that when the concentration goes to the very low level, the fluorescence signal is very, very small. That means we achieve our detection limit. And from the figure, we can tell that the detection limit for photonic crystal of IL-3 is 22.86 milligram per mil, while on glass slides, it is 205.76 kilogram per mil. So this means that our photonic crystal would allow us to use an inexpensive detection instrument to achieve even better sensitivity. So we did that to the TNF-alpha, and we determine by plotting the dose response curve, we can also determine the detection limit for TNF-alpha. And again, our photonic crystal shows lower detection limit than the glass slides. Now I will move to the second application, which where we will try to detect the microRNA on the photonic crystals. So we functionize our surface with silane, and then we add the capture sequence. And then we add the Cy5 labeled microRNA and let them hybridize. So if the sample contains microRNA, then we will see the fluorescence signal. Again, we have different concentrations of the sample, and we can plot the fluorescence signal as a function of the concentration. And from this plot, we could determine the detection limit, which is 3.6 picogram per mil. So again, this verified that our system could achieve very low detection limit to picogram per mil. So finally, I would like to summarize my talk. So here, I have demonstrated our photonic crystal enhanced fluorescence technology. In this technology, our sensor is of low cost, and also our detection system is inexpensive, and meanwhile, it can efficiently couple to PC resonance. And we also, to demonstrate the capability of our system, we also detect the protein biomarker and microRNA biomarker. And the result shows that our system could achieve picogram per mil sensitivity. Finally, I would like to thank you, the funding resources, and our collaborators. And this is our group. Thank you.
Thanks. We have time for some questions.
Very nice talk. Have you done measurements from serum or actual cell lysate yet? And would you expect a change in the detection?
Yeah, that's a good question, and that's what we want to do next. So we would like to incorporate the photonic crystal with the microfluidics, so that microfluidic channel can separate the different cells in a serum. Because if we just directly detect the serum, there are so many things in the serum, so that would greatly affect our sensitivity. Yes, thank you.
Also, how long does the whole process take in terms of incubation, washing, labeling?
Right now, so for the microarray format, it takes 3 days to finish the whole step.
On your chip, how long does it take?
Yes, because we need to incubate, and but for next step, we want to incorporate our device with the microfluidic channels, and that will allow us to have shorter assay time.
Maybe this is a related question. One of the issues is not just how sensitive the device is, but how good it is at distinguishing between different signals. Right? Do you have any sense of how good your chips are compared to the standard methods of distinguishing between different types of signals you might be looking for?
You mean selectivity?
Selectivity.
Yes. This is something that we should look at, but I don't think we have done any experiment to really see how good the selectivity. Because right now for our technology, we just have a platform to enhance the fluorescence signal. But in terms of the selectivity, that's kind of more related to the chemistry people, the surface chemistry stuff. Yeah. Mm-hmm.
Kind of a follow-up question. You said you're trying to use TNF-alpha and IL-3 to detect cancer cells.
Right.
Okay, so TNF-alpha and IL-3 are commonly expressed on multiple cell types. So how are you proposing to weed out the cancer cells from normal cells?
Okay, this is some biological question, right? Yeah. So, frankly, I don't know. Yeah, because we are more focused on the optical part right now. But that's a good point.
Those are expressed in higher level cancers.
questions? Okay, well, let's thank Yafang again, and that everyone, all the speakers, is really nice.